another video from Vishu's BioLogic. In this video, we'll talk about epistasis. So, let's get started. Now, epistasis is the process by which a gene masks or hides the effect of another gene. The gene whose effect is masked is called the hypostatic gene, and the gene which masks the effect of another gene is called the epistatic gene. Now, epistasis is of two types. First is dominant epistasis and the other is recessive epistasis. Now, we'll look into each of them separately. Now, for dominant epistasis, suppose we take example of cucurbita pepper or summer squash, which has fruits of three colors: white, yellow and green. White is dominant over the other two colors. Now, gene for white color, capital W, masks the effect of yellow color, that is capital Y. That is, in presence of capital W, yellow color is not represented, whereas green is only represented when both W and Y are in recessive form. Now, let's understand this with the help of a Punnett square. Consider capital W, capital Y as white, and there is a cross with small w, small y, that is green. So, the F1 generation will be hybrid, that is capital W, capital Y, small y, which is hybrid and is white, right? Now, if there is a self cross, that is the F2 generation, let's look at that. Then we'll have capital W, capital Y, capital W, small w, capital Y and small w, small y. Similarly, capital W, capital Y here, capital W, small y here, then small w, capital Y here, and then small w, small y here. Now, we'll just fill up the boxes. That is, this will be WW, YY, this will be WW, YY, this will be W, small w, capital Y, and this will be W, small w, capital Y, small y. Now, as I told you earlier, that wherever there will be a capital W, it will mask the effect of other genes, that is, the color will be white. So let's see which ones will be white. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, okay, 11, 12. Now, all these 12 will be of white color. Now, wherever capital W is absent, the color will be yellow. That is 1, 2, 3. So 3 are yellow. The only one which is small w, small y, that is the recessive, will be one which is of green color. Now, if you recall my previous video of Punnett square for a dihybrid cross, then the phenotypic ratio was 9:3:3:1, whereas in this it has been modified to 12:3:1 because of the epistatic gene, which is masking the effect of the other gene. Now, let's look into recessive epistasis. Here, the recessive alleles in homozygous condition mask the effect of dominant alleles. Now, let's consider the example of mice, which wild body color is greyish or agouti, which is controlled by, say, capital A, which is hypostatic to recessive allele small c. Okay? But in presence of dominant allele capital C gives rise to agouti. That is, capital C AA is agouti, whereas small c capital A is albino. Let's understand this more with the help of a Punnett square. Suppose there is a cross between capital C, small a, which is colored, and small c, capital A, which is albino. The F1 generation will be capital C, capital A, small a, which is agouti or greyish. Now, the F2 generation of this hybrid self-cross will be, here we will have capital C, capital A, capital C, small c, capital A, and small c, small a. Similarly, here also we will have capital C, capital A, capital C, small c, capital A, and small c, small a. Now, if we try to fill this box, this will be capital C, capital A, capital A. This will be capital C, capital A, small a. This will be capital C, small c, capital A, capital A. Now, wherever A is present, in presence of this dominant allele C, it will be agouti. So, 1, 2, 3, 4, 5, 6, 7, 8, 9. So, there are 9 agouti or greyish mice. Let's count the number of colored mice. 1, 2, and 3. So, there are 3 colored mice. And let's count the number of albino. 1, 2, 3, 4. So, there are 4 albino mice. Now, similarly, here also, the phenotypic ratio of the dihybrid cross has changed from 9:3:3:1 to 9:3:4. So, if you like the video, please give a big fat thumbs up. For more interesting updates and more interesting videos, stay tuned to my channel. Thanks for watching.